So hello everyone. My name is Joshua, or you can call me Josh. I'm a, PhD student in James Glacier's lab. I will be talking about today how to, I will continue a little bit of the analysis we were doing on SIR models, what type of information we can get once we know what the parameters of the model are. And then we're gonna kind of take a step back to think a little bit about how these parameters could be potentially inferred from the data. Because usually in the exercises that we do in class, we usually specify the model and specify the parameters ourselves. But in real research, these parameters are usually unknown, and they have to somehow be estimated from the data. We'll spend a little bit of time thinking about it and doing a couple of exercises. So I'm gonna hand back the presentation to James so we can discuss your projects, and then I'll be back to continue with the class after.
Okay, thank you. So why don't we just go around the room? If people want a screen share to present, that's fine. If not, just tell us where you are. Again, if you're not ready this week, that's okay, but next week we need something a little more formal. So let me start with Delaney.
Oh, it says I can't share my screen right now because he's sharing his.
Okay, thank you.
Yeah, let me, yeah, I stopped sharing.
Okay. So I chose the paper on intracellular hepatitis C modeling, and it's basically going much more into the protein functions and how the RNA replicates after someone contracts HCV. And why is it important? It's important because there's a lot of people that are still currently affected by HCV, even though we have a vaccine out. And long-term effects of hepatitis C are cirrhosis and liver cancer, and those are both fatal. And then with a better understanding of the protein function and how the RNA replicates, we can make target-specific medications and prevent long-term damage. There are two models that serve as foundations for this one, the binder and the Dahari model. They both basically assume that the hepatitis C only use cis-acting factors when replicating, because that's what was known at the time. But now current research shows that it replicates using trans-acting factors as well. So not only can the mutation show up on the same gene, they can show up on the complement as well. And this is the table of differential equations that they use for all of the rates in within the cell and within the host. And then these are my references that I had. And then I was thinking of doing a prediction on how it reacts to medication, but I'm not sure how I would do that. So that's why I said mine's a little rough right now.
TJ, you've thought about that hepatitis C situation. What, do you have any comments on that?
Yeah, I was actually going to ask, is that the compartmental HCV model that I believe Alan Perlson contributed to? Okay.
I believe so, yes. It's the one that was shared on Canvas.
Okay, yeah, it's the compartmental intracellular model. Yeah, we've actually done some preliminary work using that model in a multicellular context. So actually, I'll try not to drone on about that one, but yeah, that there's a lot of complexity to do with that model that isn't necessarily all represented just in that paper. And in fact, there's a lot to do with the the uncertainty of outcomes in infection based on some of the more spatial aspects of infection and how extracellular virus spreads throughout a tissue after it's been internalized and then subsequently released. So if you are interested at all in some of the more multicellular and tissue level components of that, I'd be happy to schedule a call sometime outside of class and we can talk about that a little more.
That would be awesome. Thank you so much.
I should say, I think that's a great paper that you picked. And I know that sometimes it seems like paper replication and reproduction maybe seems banal, but we were just on the phone with a distinguished researcher in England who works for the EMBO, the European Molecular Biology Organization. And his whole world is reproducing models, because many published models are not reproducible. And so what they do is they take papers like Alan's, they code them up in SBML compliant languages like antimony. They document them, and then they release them in a very large database called Biomodels database. And that's something that actually is a current research topic. And one of the things they're very interested in this European organization is to make it possible for people not only to have the models archived, but actually to run them. And putting them up on NanoHub makes them available. And so actually you're doing this would be part of essentially a cutting edge research, not maybe in the biology itself, although it might be, but actually in the computer science of how to distribute and validate models. And so I want to say that even though it may seem routine what you're doing, it's actually very unusual to do what you're doing, which is to take models that are published and turn them into something that anyone can use and examine. And this particular model of Alan's would be a great example of them. Many other people have come up with other good ones in the class, but this one I think is a great choice for that. Okay, thank you very much. Who would like to go next? Or shall we just go across the room? Benjamin?
Yeah, I can go. Yeah. This one. All right. So my my presentation maybe not as fancy as the previous one, but so basically I want to recreate the model from the co-infections of the respiratory tract viral competition for resources paper that I submitted as one of the ones I'm interested in. This model simulates the way that various viruses may compete for limited resources in the lungs during an infection. And I also wanted to add, because I didn't know if this was okay or not, if it is possible, because I don't know if it is, and within the scope of the assignment, I would also like to expand the model a little to include the relevant viral infection of lungs, COVID. I don't actually know right now if that would be possible because it's still a relatively new disease. I don't know if the data is out there to be able to fill in all of the needed parameters for a simulation of COVID within the lungs. The model can be seen here. It's a relatively non-complex, at least it's not a huge IDE. The target, T is the target cells, E is eclipse cells, which are a phase where the cell is infected but not yet producing viruses. I is infected cells, and V is the amount of virus. For the NanoHub application itself, I want to start with an explanation of the model and what each of the parameters represent in the biological context, as well as brief explanations of the viruses. Once that's done, I would like to show examples of what this model produces as a way of acting as a guide to read the results of the application. And then for the application itself, users will be able to select different viruses to see the results of co-infection of these viruses. And that's the full citation of the article I was referring to.
Great. Josh, you're working on co-infection. Do you have anything to say to that?
So there, I just want to say that there is already data of, there's a couple of co-infection studies on SARS-CoV-2, and at least one of the viruses that is also discussed in that paper, which is I think it was a rhinovirus. So it might be doable. It will require doing a little bit more than just replicating the results of the paper. The paper is not extremely complicated. I wouldn't expect it to be too complicated to do such an extension, especially based on what we will be discussing today.
I think the co-infection is a great topic, and if you end up showing up for the next semester of this, you could take that OD co-infection model and spatialize it, and then you'd basically be replicating the paper Josh is trying to work on right now as a research topic. So actually, these are both great problems. Okay. I guess Colin is next.
Yep, I can go. So the paper I was looking at was involved with influenza virus infection models with density dependence, focusing mostly on viral decay. And it was by Amanda Smith, David Moquin, Veronica Bernhover, and Amber Smith. So the biological relevance of this model that I found, influenza A obviously was a prevalent viral outbreak in the last few decades, and it's come with that has come many proposed models about how it spreads and how it can be fought via immune mechanisms or vaccines. This model in particular looks to analyze changes in viral decay and growth rates within the host during infection, which has not been largely analyzed in other models. It was noticed in a test run initially when a collection of mice that were infected with influenza A, they noticed the quantity of virus within the host subjects over time was not consistent, so they were measured over the course of time. And those concentration changes that occurred weren't really predictable. So this model was created to discover how those changes might look. It also allows for further analysis and accurate prediction of viral clearance from hosts over time. Some of the model specifications, they wanted to analyze the change of susceptible. There are several categories of hosts, which included susceptible cells, infected but not yet contagious, infected and contagious cells, as well as the concentration of the virus over time. A few input variables included infection rate, susceptible cells, concentration of virus, rate of transition from target to non-contagious cells, the number of non-contagious cells, and number of infected cells, along with viral clearance rate and virus production rate. And of course, the goal would be to find these specific times where viral growth and decay were the highest and lowest within the model. A few models already exist that kind of are similar and support this idea, especially those that look at viral growth and decay along with viral reproduction rates over times. One of the examples I found, or a few of them, was the localization of influenza A density within the respiratory tract, which I've linked the paper there. And then two others dealt with COVID. One dealt with the viral load functions, which analyzed different viral densities between host cells, along with a COVID-19 viral kinetics, which determined the reproductive value within COVID within a certain community. When it comes to NanoHub, an application such as this could allow users to more in-depthly observe the growth and death rates of viral outbreaks such as that of influenza A under conditions that they could preset or would be preset for them, like population size, infection rate, etc. And this would help to answer a few questions as to whether a virus would die off completely within a population or not, depending on certain preset conditions, or whether it would remain around and continue to grow. It could also help us, of course, understand what sort of requirements would need to be put in place in the real world to either stop or slow the spread of such a viral infection. And that's all I have for now.
Great. I know we had trouble connecting to talk about projects. I apologize for that.
No, that's all right.
That's Josh.
Yeah, I'm here.
Amber's on his PhD committee, so I have to kick that one to Josh.
Oh, okay.
Yeah, and there is a more recent paper from her where she actually included even more data that she did in this first one. That it might actually be more interesting. It's not terribly more complicated. It's just a little bit more realistic and uses additional data to estimate some of the parameters of the model, specifically the clearance by immune cells. So that one might be worth checking out.
Yeah, I'd be interested in that.
I think that I mean, again, I think it's a good base model. And I think it'd start with that and then see how far you can get. Maybe add on a little bit at the end if you get it done. Actually, Amber was a guest lecturer in this class, not in 2020, in spring 2020. She gave a guest lecture for us on this specific paper. So we might be able to connect you with her a little bit if that were helpful. I think these are all great topics. Again, there's such a broad variety of possible topics. I know it's not easy to pick, especially early in the class when you're, but these are good ideas. Great, thank you very much. Allie?
I don't have anything right now, but I will have something next week.
That's fine. I mean, one thing to do is if any of the things that have been presented seem interesting to you, please feel free to reach out to your classmates and talk to them about whether there's an opportunity to do something together. Because these are all projects where there's certainly projects where one person can do it, but they're projects that often benefit from having multiple people thinking about it together. One person could be assembling the experimental data or writing the explanation of the biology or thinking about good exercises to do while the other person does the coding. And there are many ways of breaking down the tasks of our project. And to really present this material well in a NanoHub app is a lot of work. And a lot of that work, some of that work is the computation, but a lot of that work is thinking about how to present the material and explain it in a way that's interesting for people and that they learn from. And I'll remind you that, again, the NanoHub apps, while they're not refereed, do give a DOI, and therefore they are publications that you can, if you're proud of, you can have future employers or universities, you put them on your CV and people see them because they're public. And so if you do a good job on them, those can be things that help you in a career establishing yourself. It's like having a portfolio if you're an artist. In this case, it's a portfolio that's online, And so it's something that's worth, it's not, one of the things I really like about the NanoHub is it's not like a term paper which disappears. It's something that is in fact out there and that can be used to advance your careers. Great. Okay, Ryan.
Sorry, yeah, I was on mute. Yeah, so I did present something last week, so I'll just start with that. Can the last person stop sharing their screen, please? Yeah, thanks. Okay. My PowerPoint seems to have run into some problem. So basically, just building on like what I presented last time, I was talking about building a neural network to kind of replicate the differential equations result of an SIR model. So I was looking at some more papers that could probably provide evidence that this kind of stuff has been done before. So one of the interesting papers that I came across is this one. It basically talks about the time window SIR model. So basically they do almost exactly what I was trying to do. They are doing this for COVID. Essentially they do it in like these 3 steps. In the first step, they are basically trying to like start with some default values for the parameters, and they are trying to predict the SIR values. Then there's a second step where they are evaluating the model parameters. So based on how the numbers of SIR, SI, and R are changing over a period of time, they are trying to kind of train the model and basically come up with values for their model parameters so that it fits the data. And in the third part, they are basically trying to predict ahead of time. So if I have probably, let's say, like 3 months of data, they are trying to predict how the SIR values will change for the 4th month based on the trained parameter values. So basically, yeah, so this is briefly how it would look like. You have your date values, you have your SIR values. You are feeding it into an LSTM model where you are trying to train the parameters for time t and predict it for time t+1. So the tricky part here would basically be updating the values of the neural network, because like since like we are going to be using ODE equations to do the back propagation and to update the values of SIR will not be like, I don't think there are custom made functions in PyTorch for that. So that will be something that will like I'll have to work on that a little bit. But yeah, this is definitely something that has been done, and there's evidence that uh, uh, there is research going on in this direction. So I think that kind of reinforces the assumption that this is something that is worthy of being looked at. And this is a very recent paper, and it's about like COVID-19. So we are trying to do it for influenza virus, influenza A virus. But yeah, the underlying principles are almost exactly the same. Thank you.
Can you hear me?
Yes.
Yes. Okay. I'm having a Zoom problem again. So that's great. I mean, potentially you could use those methods on some of the models that some of the other students are doing. So I think the thing to do is to start with a data set and a model that is established and published. But then it would be really fascinating if you could take this and apply the same method, for example, to the co-infection model, or to the to one of the other in-host models, the human hepatitis C virus, for example. So,
Yes.
so potentially it's something where we could you're doing sort of a cross-cutting methodological project that could potentially be connected to some of the other projects as well. So I think that's a great idea. TJ, do you have anything to comment on this?
No, we, I mean, I'm pretty excited about this. I think this is a nice precursor project to a lot of opportunities. I mean, this is certainly relevant work for, you know, simulation tools and models that are expensive to execute. You know, these sorts of issues are especially important when it comes to doing anything related to biology because of how stochastic these systems can be. And so being able to generate these sorts of predictions potentially with huge speed ups, I mean, has a ton of relevance.
I mean, I think this idea of sort of doing the neural network as a predictor corrector that adjusts parameters to keep the model in registration is very significant. And we've had some talks. I mean, some of the talks in the working group were on that topic. One of the early ones was about that. We might thumb through the stack of working group talks and see if there are any that are relevant.
Yeah, Ryan was showing me some interesting ones earlier today, actually, on sort of doing something to that effect, but relating those parameter corrections to more sociological factors or things sort of outside of the traditional, you know, population model.
Yeah. Like, do you want me to pull that slide up for you?
Okay, sure. I mean, it doesn't have to be at the population level because it could also be, for example, the example that we just heard where the model of Amber Smith for viral clearance within a mouse. That model is a very simple model. It's surprisingly good given the limitations of the model, but there definitely are things in the data that are not in the viral load that are not captured by the model. And if you use the same method to adjust the model parameters to keep the model in registration with the experiment, that would actually be really interesting because you'd basically be saying there's missing information in the model. And that corrector model then would be capturing some kind of mechanistic information that wasn't present in the original model.
Yeah.
You need good data sets. So the big reason that people do this with epidemiology is there's a lot of data available, high density data, high quality and high density data. It's much harder to get the same level of resolution when you're measuring the amount of virus in a mouse. It's actually surprisingly hard to measure viral loads. Their viral loads are measured in units of something called TCIDs. And the actual experimental procedure to measure how much virus, if you take a sample from the lungs and you want to know how much virus is present, it winds up, it's actually a really expensive and unpleasant experiment to do. And it's not very accurate. And so ironically, it's relatively hard to measure how much virus you have in your body at any given time. But those data sets do exist, and Amber had a lot of them. And so it's possible that you could use the same method. Since the models look similar, you could use the same method that we're talking about for epidemics to model what's going on inside an individual. It would be potentially interesting to do that. But go ahead, please.
Yeah. Yeah, thanks. Yeah. Can you see my screen?
Yes.
Okay. Yeah. So like this is essentially the paper I was talking about. It's called SIMLR. So here basically they are not using any deep learning methods. They are using machine learning. You can see that with time varying parameters of SIR, they get like pretty accurate results. It's almost spot on. So basically what they are essentially doing is that for every change in policy, they call it a CP, they call that event a CP. They are closely monitoring the parameter values and updating them. So essentially probably like something like change in mask mandates or anything like that, it gets reflected in the form of a change in parameter. So parameters like beta, gamma, they get changed at that point. Like according to them, if you do that rigorously enough, you are able to kind of land on a very accurate prediction. So yeah, again, there is no deep learning involved here, but this is also something that I think is like uh, something that can be considered. Sociological effects of the dynamics of SIR.
So what I was thinking of it for in-host modeling would be your immune response changes. Over very short times, it's the interferon response that matters to controlling the virus. And then over a period of a couple of days, it's the innate cellular immune response. It's macrophages, neutrophils, and natural killer cells. And then over the next few days, it's the CD8 plus T cells and CD4 plus T cells that come in and clean things up. And then starting about 10 days, you get antibodies that come in and clean things up. And so there are these different phases of viral infection where different players show up and do different things. And that could be, I think, conceptually a little bit like changes in policy.
Absolutely, yeah.
And they would change, essentially what they would change with the viral replication rate would change. The rate of infection of new host cells would change. The infection rate of clearance of the virus, the rate of clearance of infected cells would change. And you could build a model where you had all of those processes operating, but the rates of them were controlled by the neural network. You could see then whether you could replicate the particular patterns of viral infection that are seen. And I think that could be really an interesting thing to do. And methodologically and even model-wise, they wouldn't look very different from this. What would be different would be how you interpreted what those changes of instead of a change of policy, it would be a change of which players were operating in the immune system.
Yeah.
So I think that's something that would be worth examining. I don't know, Josh, Giuliano, or TJ, what do you think about that? I mean, doing this would already be enough, but maybe that would be even more, maybe something that could be thought about. What do you think?
Absolutely. I find that idea extremely interesting.
Yeah, but yeah, it is a lot, but super interesting. If you can find the time and the energy.
Yeah, I guess I'll start with a basic model, and if I I'll try to build on that. I'll start. Yeah.
Yep. Yeah, I find that. Yeah, taking things one step at a time and also building code in a way that it's easy to extend is a plus afterwards, even if in other stuff and projects you can reuse things and so forth.
I guess I mean, it's true for this class that may be too much, but to me, this is the kind of project that could go on.
Yep.
Could become a PhD thesis, for example. Yep. And so, I mean, frankly, all of the projects that have been talked about are great semester projects, but they could also be developed in very interesting ways. So great, that's wonderful. Thank you. And we can definitely, TJ, if we can help have Ryan look at those, look back at that list of working group speakers, because I know there were at least 2 who are using AI methods to do these kinds of predictor corrector things. Keep changing the parameter values to keep the models in registration. So we should dig through, I mean, it'd take an hour, but we just need to look, name by name through that list of, actually, that wouldn't even take an hour. Just look at the page, the IMAG page where we list all the abstracts and skim those. Because I know one of the early ones, what was his name? I think he was at Lehigh. I'm not sure. I'd have to find, maybe it was Penn State. But there was somebody who was doing something like that. I think that's fascinating. Really interesting complement to the work that other people are proposing. All right, thank you. So I guess, is it Drew? I think you're the last on the list tonight. Is that right?
See if I can screen share.
Are you there, Drew?
Oh, can you hear me?
There you are.
All right. Yeah, I need to be able to screen share.
Okay.
Let's. Okay. Is this visible? Okay.
Perfect.
All right. Well, I think I'll just quickly run through this because I still want to meet with you and talk about exactly what I want to do with this. But I think the paper I'm going to go with, I talked about last week, was this one, modeling the cell division cycle with the CDC2 and cyclin interactions. And but yeah, basically the paper is talking about how these 2 molecules are formed in the cell, and they form a NPF complex, which is, well, I mean, it forms a growth factor, and then the cell splits, and then it destroys the NPF complex, and you get this cycle that's like shown over here. And basically the model was kind of exploring how the process actually works in different states and concentrations of stuff. And they found 3 like different main modes that it would operate in, and they could like relate to like real world cell states. But yeah, so the paper talked a lot about these kinetic equations, and I think I could represent these well with tellurium. And like they've provided all the data that I would need to use. And yeah, so I think I would just want to extend this model because, I mean, it was made a long time ago in 1991. So I think I could use our tools in like Python to maybe make this model more dynamic for a user. That's what I've got.
Great. Yep. I mean, that model launched, I mean, if you look at the number of citations of that paper, it's enormous. And there are literally hundreds and hundreds of papers that extend it in various ways. And so I think, again, what Giuliano suggested, which is build that base model and get it working. And then maybe look at 1 or 2 papers of extensions that can make it a little more complicated. Complicated isn't really the goal. Complicated is by itself isn't interesting, but reveal more of the complex biology of cell cycle. One of the things that's very important in cell cycle is something called checkpointing. So cells have a whole variety of mechanisms to make sure that they don't divide when they're not doing the right thing. And that's something that could be, there are models that extend that base model to try to understand checkpointing. The cell cycle also has multiple phases. And so this basic idea of cyclins, which is cyclin A promotes the production of cyclin B, B promotes the production of C, C destroys A. And then C promotes D, D destroys B, and so on. That mixture of positive and negative feedback that you get in cell cycle winds up being a very profound aspect of how biology does things in a controlled way despite large amounts of noise and uncertainty. And those Cyclin models that John had pioneered are really are very beautiful. The simple, very simple ones like the one you showed, and then there are also ones that he's published that have 200, 300 chemical species in them. Certainly don't need to do a 300 species model for the class. And maybe you learn more from looking at the simple ones, but it's worth starting with that one and then asking the questions you're asking, which is what would be good extensions? What are good ways of learning from it? And so I think that's a great choice. And it's a little bit different from the others, which is fine too. In pre-COVID years, that would have been right spot on in terms of the topic from the typical things we do. But I think that's great. And we do use those kind of Cyclin-based models quite a lot in our own work. And so I think that's great. Okay. So everybody pretty much has an idea of something that will work. Ali, is fine to talk to people about teaming up or coming up with your own project. That's great. And now I will turn the lecture back over to Josh. And he's going to start presenting this material. And Josh, I'll warn you before I do that, I'm having another Zoom crash. And so I'm going to have to just sign out and sign back in very briefly to reset my Zoom session.
What happens with the live streaming?
It shouldn't change it at all.
Okay.
The live stream is cloud-based, so as long as there's somebody in the meeting, it should continue.
Okay, cool. Okay, yeah, those are all interesting projects. We'll see, and also I guess we are all available to help people with their respective projects. So feel free to contact any of us in case you have questions or you're running into problems. As I mentioned today, we will continue with some of the analysis we were doing on SIR models, and look what type of information, once we know what the important parameters of the model are, what type of useful information we might be able to extract from it. And and then we will, as I mentioned, we will take a step back and think a little bit more of where do these parameters come from and how do they determine the dynamics of the infection, and how those parameters could be estimated from experimental data. We'll spend a little bit of time on that, and hopefully by the end of the class, you will be able to take some experimental data, let's say on the number of cases of infected individuals, and then estimate parameters such as the basic reproductive ratio. We'll talk a little bit more about that later. So in the previous class, we discussed how the SI models can adequately describe the type of epidemics where people become eventually recover from the infection and become and stop being susceptible to the infection. There might be many reasons why that's the case, but there are diseases such as, well, when we started discussing this, we thought COVID would be one of those. It seems like it's not. COVID, a natural infection doesn't provide long-lasting immunity, at least not longer than 8 months. That's what we know so far. But there are certain other diseases for which prior exposure pretty much provides long-lasting and highly effective immunity. And SIR models are adequate to describe this type of infections. In the SIR model, you start with we call all of these compartmental models. So we have different compartments, and in the SIR model, we have 3 compartments. We have the susceptible population, we have the infectious population, and we have the recovered population. And then the rate at which people become infected are proportional to both the number of susceptible individuals and the number of infected individuals, and for example, the probability that there will be an encounter between a susceptible individual and an infected individual. And then there is also a time that it takes for an infected, or a time that an infected individual remains infectious, after which it recovers, or it could also die from the infection. We don't distinguish. SIR models do not distinguish the way in which they do not necessarily do not specify the way in which the population becomes no longer susceptible or infectious. It could be because a person died, or it could be also because it recovers and develops immunity. So the parameters of the SIR model are going to be then mu, which is the recovery rate, and then it's usually called beta, but here we sort of separate them into N and gamma, N being the number of infectious encounters between infectious and susceptible individuals, and gamma the probability that per encounter that this that an infection event will happen. Right. This is just what I was explaining up to here, and from these parameters we can calculate what the basic reproductive number is, and this basically indicates the typical number an infected individual will infect in an otherwise totally susceptible population. That's what it's called, R0. And yeah, this number has been extensively discussed in the news regarding COVID, and I don't know if you've heard what the difference between the first version or variant of COVID was, and the Delta variant. Have you? I think I heard it in 538. But have anyone has an idea of what the difference is between R0 for I think it's Alpha variant of COVID and Delta? No idea. Alpha, I think it was between, it was between 3 and 6, and Delta is like 12. So yeah, basically the Delta is 4 times more infectious than Alpha Delta. The Alpha variant is, but that information we can get from just the parameters of the model. It's gonna be what we call beta, or the infection per encounter infections rate over mu. Yeah, and that's why they say that that Delta is as infectious as mumps, for example. They've also said it in the news. So yeah, you can get a sense of how actual mathematical modeling can help you understand the severity of an epidemic. Then last time we also looked at how changing certain of the parameters of the model change the outcomes of the infection. And in particular, we saw that varying mu, again, which is a recovery rate, those doesn't necessarily affect the final number of infected individuals, nor the shape of the infection, which is sometimes important, but it does change the time scale of the infection. So for the bigger mu is, let's see, the bigger mu is the faster the disease or the epidemic is over, right? And by over, we mean that there are no remaining infectious individuals. But you can see down here that everything is changing. Everything that's changing is basically the scale on the x-axis, which means that even though the curves are the same, the time scales are different. And that's what is that. So mu is going to determine, and that you can get a sense that mu determines what the time scale of the infection is. So we also looked at the dependency of the dynamics of the epidemic based on gamma. And again, just as a reminder, gamma is the probability that an infectious event will happen per encounter. And what we do see is that gamma affects both the final number of individuals that will get infected and also the time scale of the infection. So you would expect the bigger gamma to be that the faster more people are getting infected and the more people will have been infected by the end of the epidemic. And yeah, there is an idea here that because by varying gamma and varying mu, we are effectively varying R naught. And there is an important transition when we go from R naught below 0 to an R naught above 0, right? If R naught is below 0, the infections, the infection fails to take over, and we will play with that a little bit. But as R naught approaches 1, there are big changes, or near 1, there are big changes in the number of infected individuals, or the number of individuals that become infected at the end of the epidemic. We'll see that again. We'll come back to that idea. Okay, so yeah, I guess I'm jumping. I'm talking one slide ahead of the slide that I'm intending to show. So yeah, the idea here again is that well, this is a little bit of a different idea, but the question here is how we can determine when the infection or when the epidemic is starting to decrease, right? Because at that point, we sort of know that we are at the tail end of the infection, and that the number of individuals infected at a particular time will continue to decrease. Right, so we do that by solving the IDT equation, such that that one, because that's telling you the change in the number of infected individuals. So we want that one to be negative, right? So the number of infected individuals is decreasing. And that happens only if, well, this one's a little bit better to understand. Only if the number of susceptible individuals over the initial number of susceptible individuals is smaller than 1 over R naught. And that also introduces the concept of herd immunity, because herd immunity is reached when the number of susceptible individuals in the population is such that the infection stops to, or the epidemic starts decreasing. So the idea is that we heard, so that is also an indicator. This is an important indicator. So for example, if we know what R naught is, and we know what S naught is, we sort of like have an idea of when the infection will start, or when it will reach a maximum and start decreasing once we have a particular number of susceptible individuals left in the population. This is, and that was all very conceptual, but we have an exercise. I forgot to mention, and I'm sorry, I uploaded in the lecture in the student materials folder, in the Google Drive, and in lecture 4 demos, I included exercise, or I included Jupyter notebooks for every one of the exercises that we will be discussing in class. So if you have Tellurium installed locally, then you can just launch your Jupyter notebook and use it locally. But if not, please do upload them to NanoHub, so you can sort of like walk through the exercise with me. I'm gonna give people just a couple of minutes to do that. 4 or 5 minutes should be enough. James, can you post on the chat the link to the demos folder? Okay. Yeah, so if you go to that link, you should be able to access the all of the demos for today's class. And we'll do this first one. This first one, we will simulate a SIR epidemic, and then we will calculate what the change of infectious individuals is, and then determine at what time the infection starts, the epidemic starts decreasing based on that information. So again, just gonna give people just a couple of minutes. Okay. Do people have the notebook available to them already? Let me see, I probably, yeah. Okay, cool. So the first thing we need to do is, or that we want to look at is, well, we first have to simulate the SIR model. And here in lines 5 to 18, I have specified an SIR model in tellurium. So to specify an SIR model, I write S goes to I, and I just have a parameter here, B, which stands for beta, but again, I could break down that parameter into N and gamma. And then I have I goes to R1. Here I call it K1, but this is equivalent to mu. And then I just specify some parameters. They don't necessarily have any meaning at this moment. I just wanted to have the typical shape of a SIR infection. And that's what we get here. Now, just as a reminder, once I simulate the tellurium model, I can reference each of the time series for each of the variables using a simple syntax like this. So right, that's gonna give me, well, in this case it has to be S1, right? But that's gonna give me the time series of the S1 population, and then R1 population, and so on, right? So that's just a useful handle to reference each of the time series that are plotted here.
Josh, a few people are saying they need more time here.
Oh, I don't,
So
yeah, okay, that's fine.
The poll to make sure that people are okay.
Mm-hmm. Yeah, I'm sorry, the poll was hidden. So can people please let me know when they have uploaded their Jupyter notebooks to NanoHub? So Ryan, the demos are in that link. And then I'm just asking people to either upload them or upload them to NanoHub or launch them locally if they have Tellurium installed locally. Good. And again, as I mentioned in this first part, I haven't done anything really. I have just specified the SIR model in Tellurium, shows some parameter values, and I've simulated that model to be able to see the traditional shape of an SIR epidemic. So I'm gonna relaunch just to make sure that everyone has been able to catch up. Okay, so it seems like people are catching up. So the next part, what we want to do now is, as I mentioned, we want to determine what is when the number of infected individuals starts decreasing. And to be able to do that, we need to plot this DIDT. And DID in the tellurium model, as we can see, DIDT is specified by this term minus this term in 7 minus the term in 8, line 8. And the way I can reference the rates is by the name that I've assigned to each of those rates in my equation. So see, in lines 7 and 8, I defined the equation, the rate of transition between S1 and I1 as J1, and the rate of transition between I1 and R1 as J2. So when I simulate my model, I can specify that I also want to be able to retrieve that information. So I want the rate of new infections, which is J1, and the rate of recovery, which is J2. And as I mentioned, DI over DT is just gonna be J1 minus J2. So once I simulate my model, again, I can reference the time series as using this syntax. And then DIDT, I can reference it by subtracting J1 minus J2. Okay, and then when I plot it, I see that it's positive all the way till here. And if we look back at the original model and we look at the I1 curve, what happens at that point? Anyone has a guess? So this is the derivative of this curve. What happens? Right. So the derivative is 0 when I1 reaches its maximum point, right? So I1's reached the highest number of infected individuals at a particular time. From that time forward, the number of infected individuals starts decreasing, right? So we can identify that particular time as the time in which the infection, the epidemic starts to decrease. We could potentially try to, if we look at what J looks like, so it goes negative around these 2 points, and we could potentially find the index of this value to get an idea at the time, like specific time at which the infections starts decreasing. But we have another way of doing this too, which is what I was explaining earlier. And the way of doing it is by looking at when S over S naught becomes smaller than 1 over R naught. And the way we can do that as well is we simulate the model again, right? But this time, instead of looking at J1 and J2, we just look at S1. And then based on the parameters of the model, we can calculate R naught. We already know that it's N. Let's copy this from here. This is going to be useful down here. So we know that R naught is we already know that R naught is in this case B over K1. This is what I'm calculating here. And then I am looking at when, what I'm interested in looking at is when the number of infectious individuals over the initial number of infected or the number of susceptible individuals as a fraction of the initial population is smaller than 1 over R naught. And the way I do that is that this is going to be important. It's going to show up in pretty much this lecture constantly, because what we want to do is we want to look at our Y array and determine when that array is closed, when it crosses 0. So what I need to do is to determine, this is what this line is doing, is telling me what is the index at which my S, right, I think it's Y here. One second. I just forgot to reference. Okay, so what I'm looking at is when this equation becomes smaller than 0. And that's what I'm doing here. I'm looking at what is the index at which this equation becomes smaller than 0. And then I plug that index back into my time series to get the time at which the epidemic starts decreasing. And that time happens around 5.45, which if we come back here, is around the same time at which DIDT becomes negative. And if we go up, it's also the time at which the number of infected individuals starts decreasing. Does that make sense? So this is just an important way of determining, that's a way of determining when the epidemic starts decreasing based on estimated parameters. So let's say you know what your recovery time is, you know what your R naught is, and then you know how many individuals have been already infected in your population, then you can tell whether the epidemic or when the epidemic is gonna reach its maximum and start to decrease. So again, the importance about this is that these are important, especially from a public health perspective, these are important indicators to determine what phase of an epidemic you are at. Are you on the tail end of the infection, in which case you might be willing to start, let's say, relaxing in the case of COVID, certain types of restrictions, as opposed to being in the leading end, where on the exponentially increasing end, where you might be more willing to tighten up restrictions or promoting other containment measures. Okay, so let's come back to this, to the lecture now. So as I was mentioning, these are important indicators from a public health perspective because they help orient the type of public policies that could be implemented to prevent or to curb epidemics. And there are multiple ways in which we could potentially reduce our R naught or reach herd immunity more faster. So we can potentially reduce the number of contacts between infected and susceptible individuals. And that's, for example, what lockdowns were intended to do. Of course, these do not come at, these do come with certain societal cost. So for example, lockdowns are highly effective, but they are also very expensive. And depending on the, let's say in the US, the type of the part of the country that you are, they might be harder to enforce. We can reduce the probability, even if we don't necessarily reduce the the number of contacts between individuals, we can reduce the probability that there will be a infectious event per contact. We can, we could use, for example, mask or sanitation are the type of policies that might help reduce this R naught number. And finally, we can also reduce, there's artificial ways of reducing, not artificial, but there are different ways of reducing the number of susceptible individuals that it's not just by acquiring natural immunity. Vaccinations do that. So by vaccinations do decrease this the number of susceptible individuals, depending on the type of vaccines that we're talking about and the effectiveness, but that's the idea. And by that they do reduce, they bring closer herd immunity, and they bring closer the tipping point of the infection. By the way, I have another question for you guys. I think that was discussed yesterday. Any ideas of what is the difference in likeliness of both getting hospitalized and getting the infections in the first time, getting infections for COVID if you are vaccinated versus you're unvaccinated? So again, the question is, and I think it was probably, there was some data that was discussed yesterday. So the question is, if you are vaccinated or you're unvaccinated, how many times are you more likely to be hospitalized and or to get infected by COVID? than if you are vaccinated. So I think vaccination, if you are vaccinated, you are 11 times more likely. If you're unvaccinated, you're 11 times more likely to be hospitalized than to be than if you're unvaccinated. And then if you are vaccinated, if you are unvaccinated, you are 5 times more likely to get COVID than if you are not vaccinated. And that's from data that it's been discussed yesterday. It was discussed yesterday. I'm not sure if it was by the CDC or the news outlets. But this data is important because when the vaccines were approved, they were approved based on clinical data that showed that they were 90% effective at preventing, 90 upwards, at preventing severe cases and death from COVID. But of course, that was with a slightly less aggressive variant of the virus. And we've seen that type of effectiveness has decreased a little bit with Delta becoming the more prominent strain. And also there's now some evidence that immunity might be fading, especially in elderly individuals and individuals with pre-existing conditions. But yeah, that's just to put into context some of the ideas that we're discussing here, because they are in fact related, and models can help understand how the infections, how epidemics are behaving, and what type of measures might be useful at certain times than at others. So other important indicators of the epidemic that we can get out of using, once we know the important parameters of the SIR model, is the maximum number of infected individuals. And from that and if we know what the maximum number of infected individuals are, and we know R naught, we can basically estimate what our initial susceptible population was. The mathematics to get to this equation are slightly complicated, and if you're interested, you can have a look at it in the book we're using for reference in this lecture. But we will be using this equation once we estimate some of the parameters of the model. So now we'll have a second exercise. So we'll have a second exercise where we'll calculate the maximum number of infected individuals from the model, and then estimate the number of the initial number of susceptible individuals based on that calculation. So I will be using showing exercise 4.2 now. So again, people can upload them to NanoHub. So again, what we're interested in calculating now is the maximum number of infected individuals, and using that calculation to estimate what the initial number of susceptible individuals are, given that we already have R naught. And again, this is important because in all of these discussions, we are assuming that we somehow know what R naught is, because we know what the parameters of the infection are. The problem in the real world is that we do not necessarily know what those parameters are. So we need to somehow infer them from the data. And that's what the second part of the class today is going to be about. So I'm gonna launch the poll now, just answer yes in this case if you've been able to upload this exercise 4.2 into NanoHub or launch it locally. It's just for me to get an idea that you've all have access to the notebook as well. Okay, so everyone has them. Okay, so in this exercise, what we want to do is we want to calculate the maximum number of, what is the maximum number of infected individuals, and then use that number to estimate what the initial susceptible population was. To calculate the number of infected individuals is just pretty simple. I just need to simulate my SIR model. And then I can use this NumPy max function that looks at this array, which is the number of infected individuals, and told me what is the maximum number there. And the maximum number is 400. In this model, 404 individuals. That's the maximum number of infected individuals at a particular or the maximum number of infectious individual at a single time. And then based on the equations in the PowerPoint, we can now use this information to determine what the initial number of infected individuals was by using this equation, because again, we know what R naught is because we have specified what the beta and mu parameters are. And then using that equation, I can estimate that the initial population is about 1,002 individuals. And I really started with 1,001 individuals. There might be a little bit of a disagreement because again, the simulation is not continuous, so I might actually not be getting what the maximum number is, the real maximum number is in, let's say, the model, but the simulation is close enough so I get a pretty accurate estimate of what the initial susceptible population was, which is around 1,000. Okay, I'm gonna relaunch the push poll now just to make sure that everyone was capable of running this notebook and get a sense of what we're doing to calculate S naught. Okay, so just as a recap, we've used some information from the simulation and some information from the known parameters to determine important indicators of the infection, namely the time at which the infection starts to, or the epidemic starts to decrease, And I'm sorry that I'm continuously using infection and epidemic interchangeably. It's probably because I'm so used to work with within host models that those are basically equivalent. But yeah, I do when I actually mean epidemic. But yeah, going back, we've identified important indicators of the dynamics of the epidemic, namely the time at which the epidemic starts to decrease. And we've looked at what is the maximum number of infected individuals at a single time. And we've also used some of that to estimate, let's say, unknown parameters like the susceptible population or initially susceptible population. So another important, interesting information that we can get from the parameters of the SIR model is how these parameters determine what the final number of infected or the total number of infected individuals is in the model, is in the SIR epidemic. So what we need to do to be able to do that, to simulate that or get that information, is that we need to vary our R0 parameter. And as we vary R0, we look at every single simulation. So we simulate the model for different values of R0, and then we look at the final number of recovered individuals, because the recovered individuals, every single one of the recovered individuals was an infected individual. So that tells you what the final number of infected individuals was during the epidemic. It's also important to know that we need to make sure that the epidemic is over in order to get an accurate estimate. And by epidemic is over, I mean that there are no remaining infectious individuals in the population. And we will do this together. This one is exercise 4.3. And just launching the poll just to make sure that everyone's been able to access the notebook. I'm just waiting a couple of minutes because some people are asking for a little bit more time. So. Okay, good. So so as I mentioned, what we want to do is that we want to be able to vary R0 and see what the final number of infected individual is as a function of R0. And this is what this code here is doing. I'm going to walk you through what the code is doing because it's a little bit different than the code in the presentation. Again, there's a couple of ways to do this, and I just did. This is just one form one way of doing it, but there's a different way of doing it in the presentation. James, by the way, I did want to mention that I tried using the steady state, but it had convergence problems. I don't know why, because there are certainly convergent steady state values, but I was running into the Jacobian matrix, complaining about the Jacobian matrix. So what I do first is I determine how many different R naughts I want to try, and I say I'm going to try 100 R naughts, 100 different values of R naught, and then I create a placeholder, which is basically a 2 by 100 matrix that is gonna let me, if we can see it here, store the values as I simulate the model. So basically, that's the empty matrix. It's not an empty matrix, it's a matrix of zeros. But basically, what I'll do is, as I simulate the model, I will change the values of these. In the first row, I'm gonna add in the first row, I'm gonna add the values of R naught, and then in the second row, I'm gonna add the values of the total number of recovered individuals, right? Because that's what we're interested in looking at. And then the way remember that R naught is n times gamma over mu. So I can vary R naught either by varying n times gamma or by varying mu. So in this particular case, I'm gonna be varying mu. So I created a array that has the different values of mu that I'm gonna be varying, and we can see them here. So these are all the values of mu that I will be looking at, and I chose them such that they correspond to values of R naught that go from 0 to 5. Okay. And then in the next part, I loop over every single one of these values, right? So the idea is, okay, I look at the value in this array, I load my model, and I assign the value. Oh no, I'm sorry. The one I'm changing is gamma, it's not mu. Okay, but it's the idea is the same. Yeah, I'm changing gamma, not mu, but the idea is the same, right? I either change n or gamma or mu, and that's gonna give me different values of R naught. So these are the different values of gamma. What I'll do is for each of these values, I load my model, I assign that value to the model, to the parameter G in the model, or gamma, and then I simulate the model, and I store, as I said, in the first row, I store the value of R naught, and in the second row, I store the value, the final number of recovered individuals. You can see that I'm simulating the model for a very, very long time. Here it's 1,000 time units. And the reason I'm doing that is because I want to ensure that for each of the simulations that I'm running, the epidemic has ended by the end of the simulation. And we'll see what happens if we don't do it that way. So after I do that, I can plot now the first row of my M matrix and the second row, the first row being R naught, the second row being the final number of recovered individuals. And then what we see is, can someone explain what they see here, or at least describe it? just to make the lecture a little bit more dynamic. You can write in the chat as well. So again, this curve is telling you what the final number of individuals infected, or how many individuals in your population got infected in the epidemic as a function of R0. So what happens between 0 and 1? Right. So there was no you basically have no final number of infected individuals because the pandemic failed to take over. So there were no infected individuals at the end of the simulation. There were no recovered individuals at the end of the simulation. Then what happens between, let's say, 1 and 1.5? Right. Small changes in this region, small changes in R0 result in big changes in the final number of individuals that are infected. And then from R0 bigger, let's say, than 2 and onwards, the final number of infected individuals still continues to increase, but increases at a smaller rate. And there is a point after R0, after R0 is bigger than 0, that all of your individuals will get infected at the end of, or all of your susceptible individuals will get infected at the end of the epidemic. And this is interesting because this is right. This is virus independent. This doesn't, because all the information you need is R0, right, to tell you how many individuals will get infected at the end of the simulation. Right. So basically, if that's exactly the point I was trying to make. So if with an R0 that is around 12 and a virus that can freely infect susceptible individuals, you would expect that all of your population, or at least your susceptible population, would get infected by the end of the epidemic. For a R0 between 2 and 3, which is what it was taught to the alpha variant of COVID was, you'll still expect around 85% of your population to be infected by the end of the epidemic. So just a very small changes on the R0 between after 1 in the result in big changes in the final number of infected individuals. And it also shows how important, if we were trying to break down R0, right, how we will how the dynamics of the infection would change drastically. For example, if we can, even if we don't bring it down to 0, right, that is right, but right, that is a good point, as James is mentioning. In real populations, and even in right, in real populations, individuals do not mix freely. It's not, and it's a bit so not all of the individuals might get infected, but But what I did want to mention is that let's say if we are in this region, let's say because we have somehow reduced the R naught by wearing mask, let's say, which is not the case with COVID, but it might be with influenza actually. Masks are, it seems that masks are significantly more effective at preventing influenza infections than they are with COVID, which is an important, another way of saying that we should probably be wearing masks during certain seasons of the year to prevent infections, because COVID is not the only deadly virus out there, deadly respiratory virus out there. Remember that influenza kills between 45 and 60,000 people a year in the United States. So that is a considerable number of people as well. But yeah, so around this one R naught value, even small changes can significantly impact the final number of infected individuals that we get in the population. The next thing we're interested in is, well, yeah, that's what we did. I zoomed in so I can look at the values around 1 and how they increase very rapidly between 1.1 and 1.2. So I'm just basically zooming in into this region. Zooming in here into this region. Okay. So let's do this last exercise, and then we will take a break and we will come back to do the parameter estimations and parameter fitting. So the last indicators that we are interested in looking at as a function of R naught is the maximum number of infected individuals at a single time, and at the maximum number of at the time at which that number is that maximum number is reached. And that's important, for example, because in the case of COVID, you do, if possible, you do want to know when most of your population will be infected at the same time, because that will have a downstream effect on your hospitalization capacity. So, and those are important things to do too. Remember when at the beginning of the COVID pandemic, we were constantly talking about the importance of flattening the curve, right? Because flattening the curve was not meant to indicate that we would not go through the epidemic or the pandemic, but it did mean that we wanted to reduce the number of individuals that were infected at a single time, because that had implications of how many people we could give healthcare to. So of course, these are important numbers that we definitely want to know, and we want to know how these numbers change as we change R naught. And we will do it by looking at exercise 4.4. So again, just to make sure that everyone has that notebook loaded and available to them. Okay, so in the first part, in the previous exercise, we varied the value of R. Well, it was gamma, which is because remember that R is not a parameter of the SIR model. R is something we can calculate based from the parameters of the SIR model. So if we want to change R, we need to actually change one of the parameters of the SIR model directly. And in the previous exercise, we chose to change gamma. But we changed gamma, and then we looked at the final number of recovered individuals because that was an indicator of how many people got infected during the epidemic. But now what we will look at the maximum number of infected individuals, and we will look at the time at which that maximum is reached. And this might be familiar because we were doing something similar in the previous exercises as well. So again, the first thing I do is I determine how many values of R I'm gonna be evaluating, and then I create a placeholder to just store the values of R and then the values of the maximum number of infected individuals in the second row. Then I iterate over the values of R, and notice that I do something a little bit different in this case. In line 7. In the previous exercise, I was iterating over the values of gamma. But here, I'm actually iterating directly over the values of R. And once I have the values of R, I can solve this equation to determine what the value of gamma is that will produce that value of R naught. And that's what I'm doing here. Instead of varying gamma directly, I vary R, and then using the other parameters of the model, I estimate what gamma is. I simulate the model, and then I look at what the maximum number of infected individuals is during the simulation as a fraction of the original number of susceptible individuals, just to get an idea of what is the percentage of the population that is infected, or the maximum number of the maximum. This will tell you what the maximum, what is the fraction of your susceptible population that will get infected, the maximum, the maximum number of infected individuals as a fraction of your original population. So for example, here, 0.5 means that at the highest point of the epidemic, half of the population would be infected at the same time, which is an awful number. Imagine half of the US population being infected with COVID at the same time. That would be catastrophic. I think there were estimates at some point a couple of weeks ago when we reached the peak of this last wave, where they were estimating that around, I think it was around 30 million people had COVID at the same time, which is around 10% of the US population. So yeah, and we were having, and we saw a couple of weeks ago how the capacity at hospital was reaching their limit with just 10% of the population being infected at the same time. So you can imagine how, catastrophic would it be if it was, let's say, 50% of your population or 80% of the population. So again, so coming back to this idea, I iterate over, and I know I'm talking about a lot, but I just want to put this into context to show that this is not just pure mathematics, that it also has important implications for what public policy should be taken, and what information we can extract, and we can and how we can that kind of information can be used for decision making. So I'm iterating over the values of R, and in each I'm assigning different values of gamma based on that value of R, and then after simulating, I am recording the maximum number of infected individuals at a particular time. And then after plotting, I'm sorry, I need to initialize it up here. I can plot R0 and I can plot that max number, and I can see again that there is a sharp increase in the percentage of individuals infected at a single time around 1, R0 equals 1, and that after R0 equals, let's say 12, which is what COVID, the new variant is, you could expect your at least 75% of your population to be infected at the same time, which would be around 300 million people in the US. The reason that doesn't happen is because the actual susceptible population does not match directly to the number, to the actual population in the US. Because some people are not longer susceptible because either they got the vaccines or they have acquired natural immunity and so on. But yeah, so based on this information, you can, for example, estimate, let's say you know what your R0 number is, right, again, because in real populations, not everyone is. One of the assumptions of these OD models is that the population is entirely mixed and mixing at the same time. But that is, in real populations, that is just not the case. So that is an important approximation that it's done with this type of mathematical models. That it's an assumption that goes into these models. Is important to keep in mind when interpreting the results of or the predictions of the models. The next thing that we can look at is what is the time at which this maximum is reached. And this is important too, right? Because that again is the concept of flattening the curve. We want to delay as much as possible how much the time at which this is reached. And the reason it takes longer to reach, as you increase R0, it takes longer to reach the maximum is not because you've been successfully preventing the disease, it's because actually the number is much higher, so it takes longer to reach that maximum number of infected individuals. So with a smaller R0, the epidemic, basically the epidemic ends faster, and it results in a less number of individuals being infected at the end of the epidemic, as opposed to a bigger R0, where the epidemic lasts longer, and hence a bigger number of individuals are infected at the end of the epidemic, and a maximum and a bigger fraction of the population will be infected at the peak of the epidemic. Okay, I'm sorry, I didn't mean to stop sharing. And I launched the poll once more just to make sure that everyone was able to at least run the notebook and get a sense of what it's doing. So here in number 8, it's important. This is similar to what we were doing before when we were looking at when was the array closer to 0. Now what we are getting here is the index at which the array is at its maximum. And then once we have that index, we can plot it, we can plug it into our time series to get back the time at which I reaches its maximum. That's what these two lines are doing here. These are important. These are a little bit more elaborate ideas or functions. They are all NumPy functions, but they become very handy because Otherwise, you have to iterate over the array yourself and sort of like recover what the index of the maximum is. Whereas you can use available libraries and functions to sort of like shorten the work that you can do and then focus on the important information that you're interested in retrieving. And this is going to be important for your projects later on as well. Up to now, we've used the known parameters and the dynamics of the model to sort of to determine or calculate importance in the indicators of the epidemic. When do we reach the peak of the epidemic? What is the number of infected individuals that will be infected at the peak? And how this number can vary as a function of the parameters. The problem with all of these calculations is that they are as good as your guess of what the parameters of the model are. And in real life, in real life type of epidemics, we do not know what the parameters of the model or the parameters of the epidemic are. We actually, in fact, don't even know if what we call the structure of the model is, which means that we don't necessarily know whether an SIR model is a good approximation to the epidemic or not. But in the next part, we're going to assume that we know that at least that the information that we have access to is accurately described by an can be accurately described by an SIR model. So the idea here now is that based on some information about the pandemic or the epidemic, we will try to infer or determine what the parameters of the SIR model would be. So we're going to be sort of like going backwards because that is actually how it's done in real experiments. And when James was explaining, when James was mentioning that I do spend some of my time trying to infer these parameters, that we do it in the context of within host viral infections, but the structure of the models are the same. The type of problems we encounter are similar in the sense that we try to determine what the values of the model parameter are based on the limited information that we might have from, let's say, the real world. So the first idea is that based on, let's say, we have information about the growth and the decay of the epidemic because we know the number of infected individuals at a particular instance, right? So at a particular instance, we know how many individuals are infected, which is not usually, it's not an actual easy number to get from epidemics such as COVID. Because in the case of COVID, a person can be infected and they might not know it. A person can be infected, but they might not report it. And in that case, we do not have access to the real number of infected individuals. But in this case, we will assume that it's a infection that we have a perfect knowledge of the number of infected individuals at every time during the epidemic. And if that is the case, then we can estimate the number, so the parameters of the model of of the SIR model that would describe such an epidemic by looking at the leading and tail ends of the infectious time curve. The exponential growth rate is going to be described by n times gamma minus mu, whereas the decreasing part of the infection is going to be determined by mu. Right. So this is important because remember that the number of so by looking at the, and this was important during COVID because the information you do have access to is usually first the leading curve and then the tail curve, right? After the infection has decreased, right? But just by looking at the tail, at the increasing part of the curve, you are not able, that part does not provide enough information to estimate the full parameters of the model. You need that decreasing phase to be able to estimate it, that. So what we will do first is that we will look at this tail part of the curve, at the tailing part of the curve, and we will estimate the parameter mu from that tail. Then we will look at the leading part of the curve, and because knowing mu, we can now estimate n times gamma. This is important because we cannot estimate n and gamma independently. We just estimate the product. And then once we have all of the parameters, we can estimate R naught from just looking at the active cases data. Again, we will be using artificial data, but there's, I have a application up in NanoHub that does the same as that we will be doing here, using data from the early pandemic in Italy, New Zealand, and I think it was New York. And I'll give you the link to that tool later, so you can also sort of like play with that tool to get a better sense of how the parameters are being estimated, but it's just the same workflow that we will be doing here. Okay, so in the first part, we will be estimating mu by looking at the tail of the active cases data. So we're gonna do this by, this is in exercise 4.6, or I'm sorry, 4.5. Just launching the poll again, just to make sure that everyone has the notebook available to them. Okay, so yeah, seems like everyone is with me. So in this first part of the notebook, what I'm doing is I'm simulating the SIR model, and I'm storing the results of the SIR model in these variables: active cases, uninfected, and recovered, because that's usually the way they are reported in. If you go to John Hopkins database, that's usually how this information is reported. And we will be, even though I generated this data artificially, if I were to change this with, let's say, some reactive cases in the real world, I could do the same type of workflow that I'm doing here, but instead of with this artificial data, with actual data from measured at different countries. Right. So even though the model produces all of this data, we don't usually have access to, in real world situations, we don't have access to this S1 curve because we don't usually know. Yeah, it's really hard to know the actual number of infected individuals and then how much of my population is actually susceptible. So we don't usually have access to this susceptible curve. And the R1 curve is basically a downstream, it's a downstream effect of that I1 curve. So usually we can concentrate on this active cases curve to try to estimate the parameters of the model. And again, the first parameter we're gonna be estimating is mu. And mu we can estimate by looking at the tailing edge of the curve. And we're gonna make an assumption here. And the assumption is that at the late stages of the epidemic, the rate of new infections, which is this term, is small. So we're gonna set it to 0. And once we set that to 0, we make that approximation, then we see that the rate of change of this tailing edge is gonna be described, or the rate of the decay rate of that tailing edge is described by this part of the ODE equation. And then we can solve that equation. And by solving that equation, we can see that the decay rate is determined by this parameter mu. So if we calculate the decay rate in this part of the curve, we should be able to estimate mu. And the way we estimate that decay rate is by, of course, if we look at it in a log scale, it should look like a line. And then we take the slope of that line. And by looking at the slope of that line, we can determine what mu is. And again, if the slope is not aligned when looked at in the semi-log space, then your approximation that your assumption that you're in the late stages of the epidemic might not completely apply. So you need to make sure that when you zoom into the tail end, you focus on a part where this becomes pretty much linear in a semi-log scale. And that's what I do here. I'm basically zooming in to this curve in a semi-log scale. And then I can now estimate, based on this equation, estimate mu. So what I do is that I look at, let's say I pick one value on the, let's say on an earlier part. In this particular case, I chose at time 10. And I see that the value at time 10 is 10 to the 1.9. That's what I have here. Then I pick a second value in this line because what I want to get is the slope. And I chose 18. And then I look at the value at 18, and that's 10 to the 0.2. And I feel, so I'm basically taking the slope of this curve in the semi-log space, in the semi-log scale, and I use that information to calculate mu. because again, the decay rate of the decay rate at the tailing edge is determined by my mu, and I can, by doing this estimate, by doing this calculation, I estimate mu to be 0.489, and thus the duration of the infection in a single individual is about 2 days. So an individual stays infectious for 2 days. If we look back at what the model parameter is, my mu was 0.5. So my estimate is actually quite good, even if I was just eyeballing it. Okay. So I'm gonna relaunch the poll just to make sure that everyone understood that part. So you could also try different values here. For example, you could try at 12 is about 10 to the 1.3, and at 16 is 10 to the around 0.8. You can try those values and get, can anyone try them in their notebook to see what value they get? So instead of 18 and 10, let's look at different times. So it's gonna be 16 minus 12 down here. And then in the values, it's gonna be 10 to the 1.3, and then at 16 is 10 to the 0.8. Can someone tell me what their value is when they change those in their notebooks? You can type it in the chat. So again, all you have to do is in this line, you have, because right, this is the first time point, this is a second time point. So I change it to be 16 and 12. And then here are the values. And the first value or the highest value is 10 to the 1.3. And the second value is 0.8. 1.3, 0.8, 16 and 12. I just want someone to be able to do it so you get a sense of how I'm calculating it. 0.28. So yeah, that calculation is not as good as the one I was doing earlier. And that's again, because we are pretty much eyeballing this. We're not like, we are not accurately estimating them. Still not terribly bad. Okay, so now that we have mu, we now can, yeah, okay, good. So yeah, so it's not as good as the first. And the reason I say good is because we know, because we generated the data ourselves, so we know mu, it was 0.5 in the original data. But again, we're just eyeballing it, so I wouldn't be terribly worried about it. It will be, the application I uploaded is much more systematic. So now that we have mu, we can use the leading part of the active cases curve to estimate the parameter n times gamma. And again, I want to point out that because there's a product, we cannot estimate independently n and gamma unless we have additional information. but we can estimate the product of them, which is often called beta. So what we will do is basically the same idea, but instead of looking at the tail, now we look at the beginning of the curve, which I do by plotting from day 0 to day 3.5. And then now we can use the same principle to estimate the slope, because the slope is gonna be equal to n times gamma minus mu. Okay, there was a, right. There's a typo here. It should say n times gamma, here it should be plus mu. So what we do is that we use the same principle to calculate the slope in the semilog space. In this case, I took day 3 and day 1.5, and at time 1.5, the value is 10 to the 1. At time 3, the value is 10 to the 1.8, and that gives me the slope of this curve. And then since I already know mu, now I can add mu to that slope, and that's gonna give me beta, which again is a product of n times gamma. And when I do it, I get a value of 1.71, which if we come back to the model, it was a value of 2. Again, we are approximating this. We would be much more systematic. We wouldn't leave it to our eyes if we were doing this in a serious context, but this as an educational exercise, we can do it this way. So again, I want someone to try different values now, let's say 0.5 and 2.5, and tell me what value of beta they get when they plug it into this equation. And do tell me what values of 2.5 and 0.5, and what values of the active cases you plugged in. $1.5. Okay. Anyone else? Again, remember that the ground truth in this case is 2, but we're estimating this just by roughly looking at the graph, which might not be the most accurate way of doing it. Okay. So now that we have beta and mu, we can estimate what R naught is. And once we have R naught, we can estimate what the initial number of susceptible individuals is based on the equation that we looked at earlier. And to estimate R naught, we just have to divide beta by over mu. And to estimate as S naught, we just we look at the maximum number of cases, and then we use this formula, which has R naught in it, to estimate S naught. And when we run this, we get a value of R naught of 3.5 and an estimated initial population of susceptible population of 1,100. And originally our R naught was 4 and our initial number was 1,000. So we're a bit off, but it was just an estimate that we could do quickly by looking at the curves. Okay, I'm gonna see whether everyone's caught up with me. Okay, it looks like you all are. So, right, in the previous part, we estimated this models. Wait, have I been sharing just my PowerPoint all this time? No, it was all of it. Okay, it just looked weird. So up to here, we've estimated the parameters just by calculating them from the curve. But there's alternative ways in which we can estimate these parameters actually more confidently. And we use parameter fitting algorithms that look at the data and simulate the model and try to bring the model closer to the data. And when they bring it closer, they sort of like tell you what the values of the parameters are. And there's more complicated stuff that we can do once we have estimated those. We can spend some time thinking about how much confidence we have in those values, but that is something that we will come back at in a later time. For now, what we need to do is to develop an algorithm that brings our model closer to the experimental data. So it simulates the model and then tells us, and then somehow produces parameter values that bring the model closer to the data. So the way this parameter of the most important concept in parameter optimization is the residual, and the residual is the difference that there is between a model and the data the model is supposed to account for. So let's suppose we have a model, we have some data that is some data of a species or a population that looks like this. And we say, by looking at that data, we sort of hypothesize that this can be described by a decaying equation. So the population is decaying exponentially at a particular rate. And that is described by this type of model. So once we've chose and there is just one parameter, the decay rate, that determine, well, and the initial number, but let's say we know that the initial number, that determine the shape of the curve, of the decaying curve. And for each value of that parameter, a different curve, at a different curve, the model will result as, or will be simulated into a different curve. And what we can do to quantify how close the model is to the data is to measure the distance between the model prediction and the experimental data. And we called that distance the residuals. So let's say we choose a specific value of the parameter K1, let's say 1, and that results in a model prediction that looks like this blue curve. And then we quantify how far the model is from the experimental data by measuring the distance between the model and the data, right? This idea is important. So if anyone needs me to repeat it, I can go over it again. I'm gonna launch the poll just as a way to see if anyone needs me to repeat it. You can need help or something, so just to let me know because this idea is fundamental. It's gonna be important for the rest of this lecture. Yeah, so some people do need. Okay, so again, what we are trying to do here is to quantify how far is our model prediction or our model simulation from some data that we've gathered, let's say experimentally or in the real world. And the way we do that is that we simulate the model. In this case, the model has just one parameter, K1, and it results in, it's simulated into this blue curve, which is exponentially decaying. And what we do is to measure the distance between the model and our data, is that, or how far our model is from the experimental data, is that we measure the distance between our model and each of the data points. So for example, in this case, let's suppose this is 8 and this is 6. The residual here would be 2. And here, the residual will be 1. And the residual here will be 0.5, and here it would be 0.25. So that is the residuals of my simulation. So that is the way we quantify how far the model is from the data by measuring the distance between the model prediction, which is right here, and the experimentally observed data. Okay. And then there is multiple ways we can do with those multiple transformations that we can do with those residuals to sort of like express the distance as a single number. But the most common is the what is known as the chi-squared, which is basically for each of those points, we calculate the distance between the model prediction and the experimental data. We square that distance, and then we add that distance for all of the points, for all of the comparisons. So in this particular case, again, in this particular case, let's again say the distance here is 2, 1, 0.5, and 25. That is 3.75. But what we will do is that we will square the distance and add them. So in this case, is 2 is 4, 1 is 1, 0.75 is how much is, yeah, how much is 75 by 75? But we squared that distance, 0.5 is 0.25, and we add those numbers, all of those numbers, and that gives us chi-squared, which is a single number that tells us what the difference between our model and our data is. Once we have quantified this distance, then we can use an optimizer or an optimization algorithm, which will try to reduce the distance between our model and the data by trying different values of the parameters, okay? So the whole function, the whole idea behind the optimizer is that it produces new values of the parameter, and then these values produce a model prediction that is evaluated against the data. And then the distance is passed back to the optimizer such that the optimizer can determine what next values of the parameters are produced. And what the optimizer will try to do is to try to reduce the distance between your model and the data until it cannot be reduced further. And we call that point a minimum. That is the point in the parameter space where the distance between your model and the data is being minimized. And this is gonna be, it sounds a little bit conceptual now. I promise we'll get to an exercise. The idea of this graph here is that we can think of the distance between our model and our data as a function, right? As a function that has a curve. And then the role of the optimizer is to explore different values of that function until it reaches a minimum. And there are multiple problems these optimizers can run into. I'm not gonna get into all of that detail now, but sometimes depending on the optimizer, you might get stuck in what is called a local minima, whereas there is a different parameter combination that would produce a smaller distance between your model and the data. But because of the reason the optimizer works, it's unable to get out of that local minimum. So you have different options of optimizers. Some optimizers would do what it's called local searches, which means that they are, if you have a good idea that you are near a local minima or the global minima, you might just use a local optimizer that is going to take you to the minimum distance. And that's what we will do in our exercise, because we already have a pretty good guess of what the parameter values are. So we will use a local optimizer to get us to optimize the values of our the parameters that we inferred. If, on the other case, you have no idea what the values of the parameter are, then you might need to use a optimizer that is able to search for wider ranges of parameter values and explore more to explore more combinations of parameter values. Okay, and again, this is very conceptual at this point, but we will, as we work with it, I promise it will become more obvious. The central idea here is that there is a two-way relation between the optimizer and your cost function, right? The optimizer generates the parameter values, and then you as a modeler are responsible for calculating what the cost function is, which in our previous case we explained most commonly used is a type of chi-square cost function, and then pass back to the optimizer the value of that cost function, so the optimizer can sort of guess in what part of the parameter space they are, whether they are approaching a minima, whether or whether they are in a part where it's pretty much flat. And so they have to, let's say, jump away from that region of the space. Okay, I'm gonna relaunch a poll to make sure that. And again, I know this part is pretty conceptual. It will become more obvious as we do some hands-on exercise. Okay, good. So pretty much everyone's asking me to continue. So again, what we will do is we walk through the whole exercise together. So this one is exercise 4.6. Again, we use, in this particular case, we use some artificial data by simulating the model, but we could, but in reality, we try to estimate these values from actual real data. And in the previous exercise, we've estimated all of these parameters, mu, beta, R naught, and S naught. We saw that we didn't get that good of a guess by eyeballing it. So we will use now a fitting algorithm that is will optimize the values of these parameters. The first part that we need to do is we have to define a cost function wrapper. And the idea, again, is that the cost function wrapper, and this is specific to tellurium. So the idea is that the optimizer is going to give us, the modelers, the parameter values. And we have to load, we have to be able to use those parameter parameter values to simulate the model, compare our model to the data, and give back the optimizer the distance between our model and the data. And this is what this code right here is doing. I'm not gonna go over all of it in detail because it's complicated, and we can, if you guys have questions later, we can, I would be happy to discuss them one-on-one. But again, there's 4 steps that we need to go to. We need to load our model. Then we, based on the parameters that are passed on by the optimizer, we, based on the parameters that are passed on by the optimizer, we initialize those values into our model. Then we simulate the model. And then we are gonna calculate the distance between our model and the data. And in this particular case, I'm comparing the active cases data with the active cases simulated by the model. And again, this function might be familiar because this is the chi-square function that we looked at earlier. The data minus the model prediction square, and we add them for all of the points in our data. This line here in 15, I'm not gonna explain now, I'm gonna explain next week because I'm gonna have a slide that explains it, but this is just a way of aligning my model prediction with the time of my data, the time that my data has been recorded at. But again, then I pass the square root of that difference, of the sum of the differences, and that is the distance that I want to show, that I want to pass to the optimizer. That's why this function returns the difference. In this part, I added a plot so we can actually see how far we are from the data as we optimize the model. So just so you know, we are using an optimizer library called LMFit. If you have familiarity with coding and want to have your own look at the documentation, the documentation can be found in this web page. It's explained thoroughly. You can just read. It's just 2 tabs. You read them, and you should feel quite comfortable using the library. So remember that we've already estimated the model, some of these model parameters. So now let's see how far we are from the estimated models from the actual data. And again, this is based on the library, but the library requires you to create these parameters objects. And once you've created these parameter objects, you initialize one by one the parameters that you want to estimate. In our case, we want to estimate S naught, we want to estimate beta, and we want to estimate mu. So we just initialize each of these parameters, and then we can evaluate, we can call the cost function that we defined earlier with the values of these parameters that were the values that we estimated in the previous exercise. And when we run it, we see that we were a bit off. And we knew that because we already knew what the values of the parameters are. But in reality, you don't know what the values of the parameters are. And this will be an indication, by looking at a plot like this, it's an indication that how far or close you are from your data. Okay. And then the last part I wanna do tonight is this part when we actually run the optimizer. So again, based on the LMFit documentation, we need to initialize and minimizer function, minimizer object that takes the parameters objects and the cost function as inputs. And then I use this call to actually run the minimizer, and I have to specify what methods. Remember, I was explaining earlier that some methods do local searches and some other methods do global searches. In this particular case, we're using a local search because, again, even though we don't have the exact values of the parameters, we have a pretty good guess, and we are highly confident that we are near the real values of our parameters. And then when I run that, I can see how the optimizer is simulating the model for different values of the parameter and trying to get the model as close to the data as possible. And I'm gonna relaunch the poll just to get an idea, not just that you were able to run the notebook because that's just hitting enter, but that you sort of like get a sense of what the notebook is doing. And this is just, it's important because it's an indicator for me of how much I need to go over it again next week, because we're gonna go over a little bit of this next week. So if this was not super clear, you can need help, and then next week, I know that we can go over it a little bit slower or faster, depending on how well the material was explained here. But again, you see that the optimizer was successful at bringing the model pretty close to the data. And then when we look at the results of the parameters that we get, these are now really close to the parameter values that we used when we generated this data artificially, which was 1,000, 2, and 0.5. Okay. So I'm gonna stop here now. If you guys have any questions, please feel free to ask them now. Otherwise, you can also email me or email James during the week, and again, we will go over some of these ideas at the beginning of the next lecture. And thank you everyone for participating whenever I was asking questions. And I'm gonna hand back to James.
Okay, thank you, everybody. I appreciate it. Thank you, Josh, for being willing to do that. It's a great notebook that you put together. I know that if you haven't done optimization before, some of this may be unfamiliar. There's the chapter in Herbert's textbook that's very good. Wikipedia has very good articles on it as well. And so I recommend those as resources. And another thing to do is to actually take the code that Josh shared with you and open it up, look at it line by line, run it, and play with it. Because it's optimization is partly art and partly science. There takes some time to get used to it, and it's important. It's something that you'll need going forward doing any kind of engineering. And so, just as a question, how many people in the room of the 5 people who are with us, how many people have actually done optimization in other classes? Maybe we just do a quick poll on that and ask. Ryan's done it. Who else? So you could guess. Benjamin's done it too. So some people have, some have not. If you've done it before and you're willing to help out, so Drew says he hasn't done it. So people who've done it before maybe can help a little bit to people who haven't. Think back to when you were learning it and what was complicated or hard to understand when you were learning it. Again, we've got a lot of diversity in the class, and that's a strength. So please do help each other. Otherwise, I'd like to thank everybody for joining us today. And if you have any questions or needs, email us. We'll do our best to be responsive. Otherwise, we will see you next week.